Healthcare works best when both patients and providers are informed and can collaborate to determine the best way forward. Genetic testing is an incredible asset to this process, but can also be quite complicated. Patients and families eager to learn something from the tests may find it frustrating when results are written purely for professionals and contain little they can understand on their own. At Fulgent, we deliver straightforward genetic test reports that patients from all walks of life can understand. We also ensure that genetics professionals have enough information at their fingertips to answer any detailed questions that come up. This requires a balance of simplicity and complexity, and today we're going to go over how we've approached this challenge, including some sample reports. Part 1. The basics. The first page of the report gives a complete picture of the overall results and provides some general recommendations on what to do next. At the top, you'll find basic demographic information about yourself, the ordering provider, and our lab. The QR code in the top right corner is for internal tracking purposes. If you scan it, you'll see the accession ID beginning with FT- followed by some numbers. Next up is the test performed section. This section lists the name of the panel that was ordered and how many genes were analyzed. For most tests, the full list of the genes will appear further down. We also mention what types of analyses were requested. Typically, we do gene sequencing with deletion and duplication analysis. But what does that mean? Well, the three most common types of DNA changes associated with genetic disorders are CNVs, indels, and copy number variants. Let's take a moment to go through these one by one. CNVs, or single nucleotide variants, are the simplest type of change. These are changes in just one letter of the genetic code. The first pathogenic DNA change ever identified was an CNV. It's a change of A to T at position 20 in the beta hemoglobin gene, which causes sickle cell anemia. Indels, short for insertions and deletions, are small changes that add extra nucleotides or remove them. A classic example of an indel is the delta F508 variant in the CFTR gene that is found in many people who have cystic fibrosis. In our reports, gene sequencing refers to the use of hybrid capture next-generation sequencing, or NGS, to detect CNVs and indels in protein coding parts of the genes requested. With the clinical-grade bioinformatics algorithms we apply to the data, along with manual review of the results by our genetic curation scientists and lab directors, this method alone is highly sensitive and specific. This means the results are reliable and typically don't require any more lab testing. The last category, CNV or copy number variants, are larger scale changes where whole genes or parts of genes are duplicated or missing. An interesting example of a gene where CNV are particularly important is PMP22. As all geneticists studying for their board exams should know well, this is a gene where duplications and deletions are both clinically significant but cause different conditions. Something to Google if you're curious. When your report says deletion and duplication analysis, this means we have applied our in-house CNV caller, CNV exon, to look for changes at the exon level. These are subparts of genes. The algorithm compares the amount of sequence data for the person being tested and compares it to similar data from other people tested by our lab. This method is very sensitive, able to detect CNV of 2 or more consecutive exons routinely, and even CNV of just 1 exon in special cases. Unfortunately, it's not very specific. This type of coverage depth analysis is susceptible to variability in specimen quality, random noise, and other things that can't be controlled. So when we do see a suspicious CNV called by sequencing, we follow that up with a confirmatory test before reporting it. We use qPCR or MLPA to do the confirmatory test. Part 2, results. Okay, we've made it past the first quarter of page 1. We're really going to get moving now. Thanks for sticking with me. This section is designed to convey the overall results of the test. In this example, we include a little graphic of a clipboard with an exclamation mark to indicate that a clinically significant finding is present. We then state the result in plain English. In this case, a hemizygous pathogenic variant consistent with a molecular diagnosis of a COL4A5-related condition was identified. This sentence contains a lot of important information. First off, that the variant is hemizygous means that it's located on the X chromosome in a male who only has one X chromosome. That it's pathogenic means that we've classified the variant as pathogenic using the ACMG guidelines. Finally, when we say it's consistent with a molecular diagnosis of a gene-related condition, we're being careful here because we don't always know exactly what's going on with the patient. The genetics alone are not quite enough to truly diagnose a patient. It takes a team. In this case, we've found a genetic change that we would expect would cause Alport syndrome. However, it's important that the physician check for clinical signs and symptoms of Alport syndrome, such as hearing loss, vision changes, and renal defects. We don't know exactly how this variant will cause disease in this patient, but all the evidence that we have available to us says that it will cause some kind of disease. The table shows even more detail: the gene, the mode of inheritance for conditions associated with the gene, the specific alteration identified, the zygosity, whether it's heterozygous, homozygous, or in this case hemizygous, and the lab's classifications using the ACMG-AMP guidelines for sequence variant interpretation. You'll notice there are two tables listed: clinically significant variants and additional variants of potential clinical relevance. Again, these labels have been worded very intentionally. A variant is labeled clinically significant if it is deemed to be sufficient to cause some or all of the clinical concerns for the person being tested. In this case, a hemizygous pathogenic variant was identified in a gene that causes X-linked Alport syndrome, and the person is being tested for Alport syndrome. This is clearly clinically significant. The additional variants of potential clinical relevance table includes basically everything else that doesn't fit into that first category. There are two main types of variants reported in this table: variants of unknown significance, VUS for short, and carrier variants. VUS by definition are not linked to any condition, so they definitely can go here. Carrier variants, which are heterozygous changes in recessive genes, are known or expected to cause a condition, but the person being tested has a second copy of the gene that appears to be okay, so we would not expect them to be affected. If we find two different heterozygous pathogenic changes in the same recessive gene, that would typically bump it up to the first table. Sometimes we include an additional table called the supplemental table. Typically, we'll include variants in that table if we think they are very unlikely to be of any clinical utility, but may be interesting to report for other reasons. Examples of this could include VUS in recessive genes, or familial variants which are classified as VUS or benign that we don't think are clinically relevant, but the provider has asked us to look into. Part 3. Interpretation. The last section, typically appearing on page 1 of our report, is the beginning of the interpretation section. In notes and recommendations, we provide a mix of standard language and test-specific information we feel might be relevant for the case. In addition to recommending genetic counseling, we may note if familial testing is recommended for VUS, or if there are any other concerns we feel are worth sharing. We also recommend that the care providers review this information in context. At the lab, we don't always have access to detailed information about the person's medical or family history that may be relevant to interpreting the genetic results. For this case, knowing that the person had clinical signs of Alport syndrome made the interpretation very clear. If we had seen the same result in an adult with no ocular, auditory, or renal concerns, we might have come to a different conclusion. As much as we'd like to be able to do this work entirely in a vacuum, nature doesn't always conform to our expectations. In many ways, genetics is a field defined by exceptions more than rules. So we think of each test as a partnership between the person, the provider, and us. Now is when we start to dive a little deeper. The remainder of the interpretation section is where you can find the nitty-gritty details about the genetic variants reported. We include a general section about the gene that a variant is in. This is typically a paragraph of information regarding the types of genetic conditions associated with that gene. The links will take you to primary or secondary sources like PubMed and OMIM, which are bedrock tools used by the entire genetics community. Next up is the variant table. We've organized each bit of information we might think is relevant into bullet points and then grouped those bullet points into categories. First up is zygosity and inheritance. This section describes the state of the variant, typically heterozygous, homozygous, or hemizygous. This section would also include the origin of the variant if parents were tested as part of a duo or trio. Now it's probably a good time to mention that we offer duo or trio testing at no additional charge for all of our panels. This means that if you have access to parents and they are willing and interested in participating in the genetic study, we can accept their specimen and run them alongside the patient so we can know where each variant is coming from. Because this is so valuable for interpreting the genetic test results, we offer this at no charge. If the parents are not available at the original time of testing, their samples can be sent in later to do familial testing, which has the same impact. The variant type section includes the genomic location of the variant, as well as how it is expected to impact the protein. In this case, the change introduces a premature termination codon, and it is also located in an important functional domain. The variant in cases section is where we summarize any scientific literature published supporting an association between the specific variant and a condition. Here we've made an apparently novel discovery. This particular variant hasn't been reported before, but others that are very similar have been. Again, the blue links will lead you toward the primary sources. If you are looking at a printout, or for any other reason cannot access the link, you can also try a web search or go to PubMed directly and search for those numbers. The variant in controls section lists the frequency of the variant in people based on publicly available genomic data. We primarily use the Broad Institute's Nomad database, which has data for over 150,000 individuals. This is a wonderful resource that powers much of what we do. Big thanks to the team at Nomad for their major contributions to our field and for making this a public, free resource. Sometimes we may also include an additional Other Information section. This is where we would put things like evolutionary conservation, functional studies, or anything else that doesn't fit well into the other categories. The remainder of the report is technical, containing information about how the test was performed and what the test can and can't do. I'll leave those topics for another day, or you can ask us if you have questions. Try using the chat feature on our website at fulgentgenetics.com. Our team is very responsive and we're here to support you. Lastly, I want to go over a special report type. Hereditary cancer predisposition testing was one of the first areas we focused on at Fulgent. Based on our experience with clinics and patients from around the world, we've honed in on a report that is very similar to what I've shown you before, but with some key differences. First, we typically include a summary of any provided personal or family history of cancer. This information is highly valuable to keep in mind when reviewing tests, so we put it at the top of page 1. Because hereditary cancer predisposition is so complex, a brief review about the nature of cancer risk for any positive genes is included. For example, both BRCA1 and BRIP1 are associated with increased ovarian cancer risk, but of those two, only BRCA1 is also associated with breast cancer risk. The cancer risk and cancer spectrum sections on our hereditary cancer reports help to explain what a positive test result means in direct fashion. For medical management, we always recommend referring to the NCCN guidelines and oncology specialists. That concludes the basics of our clinical report. By now, you should have an easier time understanding your report from Fulgent. Let us know if this was helpful to you, and please contact us if you still have questions. We'll be posting more videos like this in the future. Tell us if there are any topics you'd like to see covered. Until then, shine brightly, everyone.